You're already ready to go.
One pole 3-component Kabachink fields. Synthesis of alpha amino phosphonate oxidizers using iron trifluoride. أويس فوزي أويس مروة الحسيني نجلاء فتحي السيد Department of Organometallic and Organometalloid Chemistry National Research Center Giza Egypt and Marwa Ahmed Fuad Pharmaceutical Chemistry Department Faculty of Pharmacy Cairo University. Introduction. Kabachink reaction is one pot reaction, 3-component. It is a reaction between carbonyl compound and amine and dialkyl phosphide, optimized condition, to form alpha amino phosphonate. Alpha amino phosphonate is a very structure which has biology and pharmacological characteristic as they exhibit antiviral, anti-tumor, antibacterial activity. They are significantly inhibit growth of human tumors. Alpha amino phosphonate containing oxidizole 1,3,4 oxidizole are significant class of heterocycles which has medicinal and pharmaceutical characteristics. 1,3,4 oxidizole nucleus are reported to induce apoptosis either through induction of caspase activity or through p53 mediated pathway. Owing the biological value of oxidizole and amino phosphonate group, the aim of this study to synthesize multifunction caspase dependent apoptotic inducer. Results and discussion. The optimal experimental design. The optimal experimental design, it is a design to refine and optimize the reaction condition. The reaction occurs between aldehydes, dialkyl phosphides, and acetohydrazide derivatives in dichloromethane. Without catalyst, we can obtain alpha amino phosphonate without in traces. So we can use this design. Alpha amino phosphonate was obtained in traces even after reflux. So we can choose two factors, parameter, catalyst, iron trichloride, iron triflate, copper triflate, and solvent, tetrahydrofuran, dichloromethane, and dichloroethane. Experimental plane of the optimal design shows that first experiment we can use copper triflate in tetrahydrofuran and iron trichloride in dichloromethane and iron chloride in dichloromethane and so on. After making and performing these experiments, we can notice that in presence of iron triflate in dichloromethane, we obtain a high yield in short time. As can be observed, it can be pointed out that using iron triflate or copper triflate as catalyst and dichloromethane or dichloroethane as solvent generally afforded compound in moderate to high percent yield, and lowest yield was obtained when using iron trichloride. Once the fitted models were validated, the optimum condition will be determined using numeral optimization by maximize the percent yield and minimize the time. The solution obtained were the catalyst iron triflate and the solvent dichloromethane with desirability value of 1. This means that using iron triflate in dichloroethane and at reflux temperature induce the highest catalytic activity, highest percentage in short time. So we perform our reaction, aldehyde and acetohydrazide derivative and dialkyl phosphide in iron triflate, dichloroethane at reflux temperature to form 13 aminophosphonate derivatives by using varieties of aldehyde to study the structural relationship. The mechanism of this reaction: first, condensation between aldehyde and amine to form imine structure. Then, attack of dialkyl phosphide to C double bond N to form aminophosphonate. Biological activity: in vitro anti-fluoritative activity and selectivity. This table shows IC50 of synthesized compound against the 3 cancer cell lines: colon cancer cells, breast cancer cells, liver cancer cells. The results show that the aminophosphonate, which carry naphthyl moiety and chlorobenzene moiety, show IC50 9.5 and 9.2 compared to doxypyroxene, which is IC50 9.4. MC7 cell line, our structure show moderate activity to two cell lines. And liver cancer cell, effect of most cytotoxic activity structure on normal cell. One of the major constraints of anti-tumor drug is absence of selectivity between malignant and non-malignant cell. We therefore assess the most cytotoxic activity structure on normal colon cell. This table shows that they are active and selective to malignant cell. Structure activity relationship study. In colon cancer cells, the anti-tumor activity of aminophosphonate derivative pairing unsubstituted phenyl group was significantly increased by substituting with cyano group in paraposition. And chloro derivative is the most cytotoxic than fluor and brom. Furthermore, also substituted more than para-substituted, and di-substituted is more than mono-substituted. In breast cancer cell, generally either substituted or replacement of the phenyl ring, significantly enhancing its activity. In both breast cancer cells and liver cancer cells, cyanophenyl derivative and 2-cyanide derivative show highest activity toward the 2 cell line. Apoptotic assay. The most effective compound were therefore screened for their prospective ability to initiate apoptosis cascade in colon cancer cell. Caspase activity increase in the level of active caspase 3, 8, and 9. Cytochrome C assay overexpress cytochrome C. And good induction of PAX and downregulation of BCL2. Caspase 8 and cytochrome C initiate caspase 3, and caspase 3 initiate caspase 8 and 9 initiate the caspase 3. So it cause the cell death. They should but show downregulation of BCL2 protein concentration. The most active structure towards colon cell line. Effect on PAX and cytochrome C, and in sequence caspase 3 and caspase 8, which follow caspase 3 to cause apoptosis. Apoptosis is programmed cell death. Apoptosis study and cell cycle analysis by flow cytometry. According to the above the results, it can be noted that compound and our compound demonstrate pre-G1 and G2M cell cycle blocking. It also evidence that compounds pairing to chlorophenyl moiety and naphthyl group are mainly antitoxic, producing programmed cell death. Conclusion: one pot 3 component Kabachink fields was optimized and successfully utilized as an efficient synthesis of new alpha aminophosphonate derivative, pairing oxadiazole moiety using iron triphenate as an excellent catalyst. Compounds pairing to chlorophenyl moiety and 2-naphthyl group show promising anti-fluoritative activity against colon tumor cell, comparable to doxycycline. The selectivity index of them were also calculated to evaluate their safety. These compounds were found to increase the level of caspase 3 and increase the levels of caspase 8 and 9, and overexpression of cytochrome C and PAX-C and PAC, and downregulation of BCL2. Thank you.
Okay, we have plenty of time for questions. Any questions? Any questions? Any comments? From the speakers, any comments? No? Yes? Well, great. Thank you so much, everyone. If there are no questions for any of the speakers, I guess we're finished. So thank you again. They were pretty informative.